And here we are. All right. So I understand that there are many of you that may not find a chapter on fungi to be the absolute greatest thing that's happened to you, even this week, right? And this week just started yesterday. I understand that, but what I want you to know is that fungi are just, they're such an important group of eukaryotes that I just can't help but to pull a lot of test questions from this. I mean, I can't help it. So while I know that you may not be motivated out of a strong desire to learn everything about fungi that you can possibly learn, I hope that you could be motivated to learn about fungi, knowing that learning as much as you can be a tool to help you completely change your fate after exam one. Okay? Completely change your fate. And I hope you all enjoyed 100% Quiz Friday. It felt really good to just put a 12.5 in for everybody because everybody was here. I mean, literally everybody got it. It felt so good, in fact, that I think I'll do it again, not this week, but it will happen at some point again this semester. I won't tell you when because I want it to be a surprise, you know, and I don't want you to expect it, but it will happen again. All right. So framing questions. Again, we're going to try, if we can, to get ahead a little bit. So I'm going to do my best to get us through the first 3 framing questions today, which should get us through the second 3 on Wednesday and allow us to start talking about animals on Friday. All right. So first question, what makes for a successful reproductive strategy? And this comes out of your chapter on fungi, because this is what's interesting. So this chapter comes before the chapters on plants, right? This is chapter 24. The plants are chapters 25 and 26, I think. Not 27, because 27 is where we start animals. And so this chapter devotes a large section to talking about reproduction in eukaryotes. And we've already talked about some of this in plants, but that's because we did something the textbook didn't want us to do. And we skipped this chapter, and then we came back to it. But I think it's helpful to just see some of this. So some of this will be a reminder of what we've talked about already, but it's nice to think about that this is true of not just fungi. This is true of all organisms. This is what makes a successful reproductive strategy, and then we'll make it specific to fungi. So this you should read and be like, well duh, right? That reproduction is only one of the characteristics of life, right? What are those characteristics of life again? What do living things do, or what do living things have that non-living things don't do and non-living things don't usually have? Metabolism, right? So that is, they process chemicals and they make other chemicals, right? So living things do that, non-living things usually don't do that. What else? Homeostasis, right? So living organisms maintain internal conditions, non-living organisms don't. Hayden.
They have DNA.
They have DNA, right? And viruses sometimes have DNA, but non-living things usually don't have DNA. What else? Make copies of themselves, right? And again, that's just one of the characteristics of life. But it is essential. And this, it says it's essential due to senescence. Senescence is loss of cellular function. Okay, senescence is loss of cellular function. So because a cell cannot live forever, cells have to reproduce themselves and basically, in some ways, start the process over, start the clock over. So when I was in high school, there was something called the death clock, and it was crazy popular. You would go in and you would put factors about yourself and your family, and it would tell you exactly when you were going to die. Told you exactly at what age, and then you could purchase a little clock that you could set like on your end table or anywhere you wanted to in your house on the wall, and it just counted down. So like it started with like however many years, months, days, and hours and minutes and you had left, and then it would count down to 0. It was awesome. It was awesome. It was hugely popular when I was in high school, and it essentially explains why reproduction has to happen, because everything has a death clock, right? You may not know when that death clock's going to come to zero, but every living thing has a death clock. And at some point that clock's going to go to zero, and so you have to restart the clock every now and again, and you do that by reproducing, by making new cells. Are you looking it up?
Yeah,
I'm sure. I mean, it was wildly popular when I was in high school. It's not as popular now. People are like, wow, that's weird and creepy. But when I was in high school, this is how we thought. Like, this was just awesome. Like, some people that I was in high school with would wear one around their neck. They'd sell necklaces, and it would have your clock. And you could just take it everywhere with you. It's fantastic. So, according to the principle of allocation that we talked about last week, individuals only have a certain amount of energy to invest in all of life's activities. If reproduction is just one of those activities, it means any energy you invest in reproduction comes with a cost, that now you can't invest that energy in something else. So what that means is you have to reproduce because no cell's going to last forever, but the act of reproducing is actually going to shorten the lifespan of the cell even more. Right? Because if you're going to invest that energy in reproduction and making new cells, you're not going to be able to invest that energy in foregoing or fighting off senescence. Okay? So you have to reproduce because of senescence, but reproduction makes senescence come sooner. Right? It's kind of a fun thing to think about. Right? So senescence actually takes time away off of your death clock. On top of that, organisms tend to produce as many offspring as they can. And that's not like just let's make 100,000 offspring like a fruit fly does, but let's try to maximize the number of offspring that survive to make offspring themselves, right? And so there are different strategies. Maybe you just produce as many offspring as physically possible, and then you don't care about how easily and how well they survive. Or maybe you produce very few offspring, but then you invest in their survival, right? Produce very few offspring, but invest in their survival. But anyways, if every organism is trying to maximize their reproductive output, what you're going to get is more total offspring than can possibly survive, right? And so that's going to fuel natural selection. Not every individual can survive and reproduce, so only the individuals that do so best, that survive best and reproduce best, are going to actually leave a meaningful impact on the gene pool. So now let's actually address this question. Okay, so that makes sense, but what makes for a successful reproductive strategy? Well, successful individuals, the animals or fungi or plants or other types of eukaryotes or even bacteria, those that make the biggest impact on the gene pool, okay, those that produce the most offspring that survive to reproduce themselves, tend to reproduce both asexually and sexually. They reproduce asexually when the conditions are favorable and they can just make a whole bunch of copies of themselves, and you're just trying to maximize the number of you that exist, right? You're like, I got this, like this environment is totally my thing, and so I'm just gonna make a whole bunch of clones of myself, and this environment's gonna be their thing too, because they're genetically identical to me, right? But then you're still capable of reproducing sexually when the environment is changing. When you're like, hey, this environment is not my thing, but maybe I can produce offspring that have some of my features, but have some features from someone else, and so that environment could be their thing. And they could be more successful at living in this environment than I am, right? And so this is a really successful reproductive strategy, being able to reproduce asexually when the environment is favorable and you can make a whole bunch of copies of yourself really quickly, but also able to reproduce sexually when the environment is dynamic and it's shifting to potentially get some offspring that have unique features when compared to you and maybe greater survival than you have. So most fungi produce both asexually and sexually. Only one phylum of fungi lacks sexual reproduction. We'll talk about which one that is because you all love when we just do taxonomy, right? And we're just like, this phylum, this is what's true of that, this phylum, this is what's true of that. That's your favorite part of this class. So I will not prevent you from experiencing that from chapter 24. Yeah, Emerson.
So is it that some plants are asexual and some plants are
This is fungi.
I mean, some fungi? Or are they both in themselves?
Most fungi can produce both asexually and sexually. There are some fungi species that only reproduce asexually, that do not reproduce sexually. All right. So, but the norm is for a fungus to be haploid, and that's a term you've seen before, right? What does it mean? That is a good question, Chris says. What does haploid mean, Allison? One copy of the genome. So most fungi are haploid. So how can a haploid cell participate in sexual reproduction? Because if you're going to take your copy of the genome, mix it with another individual's copy of the genome, now you have a cell that has 2 copies of the genome. But that is not the norm. So this is how fungi deal with that. That they produce after the zygote forms. So after one individual reproduces with another individual fungus, and you form a zygote that has 2 copies of the genome, it goes through meiosis to regenerate a haploid cell that will then develop into the adult form of that fungus. Have we seen something like this? The answer is yes. The question is where? Plants, remember, have that alternation of generations, so they at least have some significant experience as a diploid individual. It may not be the dominant form, right? So in bryophytes, the gametophyte is the dominant form, but at least has some kind of experience as a sporophyte in a diploid state. So we have seen it before, not in plants. Yeah. In green algae, or green algae if you prefer. Okay? This is kind of the norm for green algae. They live their lives basically as haploid, but they reproduce sexually, forming a diploid cell that basically immediately goes through meiosis to regenerate the haploid state. All right? You love it, right? You're like, man, I didn't know I was going to love fungi so much other than on my pizza, but this is awesome. No? You don't like mushrooms on your pizza? What's wrong with you? It's terrible. I bet you like pineapple on your pizza. It's disgusting. Actually, it's good too. I mean, it's pizza. I'll take anything. All right. So here's like a typical fungal life cycle. Okay, typical fungal life cycle. Here's the fungus. Look at that. One dark spot illustrating it has a single nucleus and a single copy of the genome. The kind of fun thing about fungi, this is bonus knowledge, which means that you won't be assessed on it, but usually the diploid condition, the nuclei stay separate. One nucleus has one copy of the genome, the other nucleus has the other copy of the genome, but they don't fuse together, which is unusual. That's very unusual. That's bonus information though. So this is a typical fungus, rockin' life in its haploid state, and then here, just reproducing asexually when environmental conditions are favorable and it wants a lot of copies of itself. Okay? And then here, there's something shifts, like the environment starts to change. This will oftentimes happen in fungi that live in temperate areas, where it starts to get really cold at a certain time of year. And so it'll shift and they'll start reproducing sexually. And here, now you have a stage where, look at that, one nucleus, 2 nuclei. One nucleus, 2 nuclei. Still separate, both with one copy of the genome. And here we've got a zygote. Very quickly, you'll form that diploid cell and then goes through meiosis to restore the haploid condition that then grows into the fungus. Right? This is a typical fungal life cycle. Allison.
So aside from the fact that there's more variety of genetic information, are the spores different when produced sexually than asexually, like actual?
Yes.
Spore?
Yes. Yeah, because you have asexual spores and you have sexual spores. Yeah, and fungi usually produce both. The spores are structurally and they are structurally developed to facilitate movement, dispersal. And so that's the function of spores is for dispersal, to make it to where the next generation doesn't compete with this fungus for nutrients. And that's why, you know, fungi will release spores into the air, you breathe them in, the fungus will start to grow in your lungs. It's just wonderful, right? And it's all because of how fungi reproduce. Now, in order for meiosis to happen, the nuclei have to fuse in order to separate them out, but they fuse just long enough for then meiosis to happen. But sometimes and we'll see this in some fungal groups, where they may live in this heterokaryotic state where they have two nuclei for long periods of time. And so then it's like, is this like alternation of generations, haploid and diploid? Or is it something completely different? So here's a typical fungus. A couple of terms I want to illustrate here. So here are hyphae. This typically, the fungal structure, no matter what type of fungus it is, unless it's single-celled, we'll talk about that. But most multicellular fungi are composed of these strands that we call hyphae. Okay? And by we, I mean you need to call them hyphae as well, right? You can't just be like, oh, we, like Dr. Ingo and all the other weird people that think about fungi, call them hyphae. No, we all call them hyphae, okay? And so if you're going to form a reproductive structure, like right here, we have our sporangium, which is a fancy way of saying spore-producing structure. Okay, sporangium. You actually have to have hyphae combined together to form these complex structures. So it's another just norm and feature that is common in fungi. All right. Any questions? Did I already tell you all Happy Monday? I wanted it to be on the recording. I knew I already told you Happy Monday, but I wanted, if you go and you listen back to this, you're like, wow, Dr. Ingo, he's so kind. Keeps wishing me Happy Monday. And then at the same time, you'll look at your grade on the exam, and you're like, Dr. Ingo, he's so evil. Right. All right, so what features do fungi share to make them unique? So far, we've just talked about fungal reproduction, and you're like, wow, they reproduce in some ways similar to green algae. There's some, unique differences, but what are some unique fungal features? I'm glad you asked. Glad you asked. First of all, fungi have cell walls made from chitin. Fungi have cell walls made from chitin. We've seen cell walls before, right? What organisms have cell walls? Plants have cell walls. Their cell walls are made of cellulose. Bacteria oftentimes have cell walls. Their cell walls are made of peptidoglycan. Archaea have cell walls. Their cell walls are made up of all sorts of different types of things. Okay? They're extremophiles, right? They use whatever's, they're like the doomsday preppers, right? They just use whatever they have available. Wow, nothing on that? That was hilarious. That was so funny. All right, so cell walls, fungi have cell walls made of chitin. You know the only place you really the only other place that you usually find chitin is in the exoskeleton of insects and crustaceans. And here are fungi just rocking cell walls made out of this material. It's weird. Fungi, they have plasma membranes, so do bacteria and all eukaryotes, right? This is a characteristic of living organisms, having a plasma membrane. But fungal plasma membranes are stabilized with something relatively unique. It's a unique type of lipid called ergosterol. You're like, man, I had no idea that fungi were so interesting. I already knew they were delicious, but I didn't know they were so interesting. Fungi digest material external to the cells. This is really cool. Although I know what you're thinking, well, don't we do that? Doesn't our chemical digestion happen inside of the stomach and the intestines outside the cell? It does, but this is something unique from organisms we've seen so far.
What does that mean? Like they digest it externally?
Externally, so the material is not digested inside cells, but instead it happens external to the cells. So they'll secrete digestive enzymes that will break down the material, and then the cells will drink up the little bits. It's one thing that makes fungi so great at decomposing materials. Like a tree that fell down in the middle of a forest, or a body of, I don't know, like a coyote, left in a moist place. Multicellular fungi as opposed to yeast. Yeast is a general term. Now there are specific types of yeast, right? Like the yeast you put into your dough to make pizza, right? Or into your dough to make a loaf of bread, or into a fluid to brew some kind of alcoholic beverage. That's a specific type of yeast, but yeast is a general term that refers to a single-celled fungus. Single-celled fungus. Anyways, multicellular fungi, they have two stages, and these are unique stages. They have the vegetative stage, where those hyphae are just thread-like, spread out. And then they have the reproductive stage, where your hyphae fuse together to form a reproductive structure known as the mycelium. The sporangium could be an example of that if it's producing spores. It could be an antheridium if it's producing male gametes, or an archegonium if it's producing female gametes. We've seen those terms before, right? In plants. Antheridium, archegonium. We have seen those before. We've seen them before. Yeah.
Okay, so it's called the mycelium?
Is a general term that means reproductive structure. So it could be a sporangium if it's producing spores. It could be an antheridium if it's producing male gametes. It could be an archegonium if it's producing female gametes. But the general term is mycelium, a general reproductive structure. All right, lecture break? Lecture break, okay. So I want you to, thinking of these features of fungi and anything else you can think of about fungi, I want you to explain to those around you and to me why fungi are in the same drawer of eukaryotes as animals. Why are they in the same drawer? Right? You know what I'm talking about, drawer? We've got this file cabinet with 6 drawers, right? Our 6 supergroups of, our 6 supergroups of eukaryotes. Thank you. All right. So why are they in the same drawer? All right. Take a couple of minutes, meaning 2, starting now.
Why do you think so?
I think it probably has to do with the fact that they they're not
Oh yeah, all living things do.
because a lot of times I found in dark places. And so they're going in a dark place, light can't reach them, so that's why they have the ability to use like other materials.
That makes sense. Yeah.
So, because a lot of times, like, since you don't have to find mold in it, you usually find mold in the dark place.
Have about 45 seconds. Keep the convo going. Shovel some more coal in there.
You need to have the right time.
Man, first one.
That's like it's really because this one right here is the key one that plans and because they all get to do
Alrighty. So we're going to do something a little bit different because it's Monday of week 6. During our discussion, everybody's standing. Everybody's standing up out of your seat. Hey, don't give me the thumbs down. Oh, we're all standing. You too, Morgan. And Mackenzie. Oh, yeah. Just until we finish our discussion, we're all standing. Wow, this is fun. I can still see everybody. This is great. All right. So why would we put these organisms in the same drawer of eukaryotes as animals? Yeah, Amanda.
It has. I think it has to do with how they gain their nutrients. Is that the fungi have to like get their nutrients from another source of material, whereas plants can produce their own nutrients through photosynthesis.
Right. So we have several of those drawers that are entirely photosynthetic. Right. You've got Archaeplastida, entirely photosynthetic. Every member of that drawer. You've got Chrome aviolata, almost entirely photosynthetic, although we still have 3 other drawers that animals and fungi aren't in that have quite a few heterotrophic forms, but they do share certainly share some aspects of their life history. Emerson and then Autumn and then Hayden.
Like animals, the fungi reproduce sexually.
Okay. That's true. Yep, they for the most part, most fungi are capable of reproducing sexually. Although that tends to be true of most eukaryotes that are capable of some level of sexual reproduction. Now, their sexual reproduction in fungi is actually fairly similar to green algae, but does have some similarities with how meiosis is carried out to other. It was Autumn and then Hayden, right? Okay. Heterotrophs? Yeah. Hayden.
Well, plants have chloroplasts, fungi, and animals don't have.
That's true. Yep, they are. So they are, basically, remember, you could put your eukaryotes into one of 6 drawers. There's another way to organize them, right? You can organize them into 4 groups. What are those 4 groups? Plants, animals, fungi, everything else. So you're like, we can figure out what a fungus is, unlike the everything else, right? That's like the miscellaneous drawer. And it's definitely not a plant. Definitely not a plant. That's good. Yeah, Levi.
Is it because they kind of use a similar digestion process of putting out enzymes and breaking things down?
That is true. They do digest material extracellularly, which tends to be unique among eukaryotes. Remember, the Venus flytrap does something similar. Somebody brought up a Venus flytrap, and then Calvin, was it you that asked if a vegetarian ate one? Would they still be a vegetarian? I was thinking about that this morning, and I told you yes, but I don't know why, and I'm a little torn. Like, I don't even know. And then there are like, anyways, I mean, sorry. Anyone remember what the drawer is called? We'll see it in just a moment here. It's at the bottom of this slide. Opisthoconta. Oh man, what on earth does that mean? Stay tuned. Emily.
I just have a quick question.
Yeah.
So, digesting extracellularly, how is that related to animals?
Because that's how animals digest as well. Animals digest extracellularly.
It's like even though it's not the same, as you don't think of it in terms of our body, but if you actually think about it in our stomach is just this big open space full of unlike the acids that is produced by the cells that are lining the stomach.
Yeah. Yeah. All right. Cool. You all can sit down now. Thank you for humoring me. Hopefully that will help you stay focused for the remaining 19 minutes we have. Yeah, Allison.
I was just thinking about the vegetarian.
Yeah.
All the plants help decompose organic material, right? So I don't know if that's
I mean, I'm convinced that you can eat a Venus flytrap and still be a vegetarian, but it's such a unique type of plant. I mean, it's certainly a carnivorous plant. And then but I guess if you were going to say, well, that's like eating an animal, then you're like, well, what if I only eat beef? Like, I don't eat any carnivorous animals, like I'm not eating bears or raccoons or weasels or squirrels,
It's a
right?
vegetarian couch, a vegetarian steak.
Yeah, yeah, Tara.
Could it be like you're still a vegetarian, but you're definitely not vegan?
Oh man, I don't even know. All right, so now let's start talking about some features that they do share with animals. Somebody mentioned that they lack chloroplasts. That's right here on this slide. That was Hayden? Yeah. Nicely done. Store excess sugar as glycogen, as opposed to how do plants and algae store excess sugar? As starch. Okay? So they store their excess sugar as glycogen, and then they're part of Opisthokonta. Part of Opisthokonta. And this term refers to the structure and the function of flagella. And I know what you're thinking. You're like, but the sperm of bryophytes and vascular seedless plants, they have flagellated sperm, and so do green algae. And you're right. But that's what this group is referring to, the presence and the particular type of flagella. But basically, to answer this question, why are they grouped in the same drawer? And it's almost entirely due to genetic similarities. That animals, why didn't you mention that?
Oh, I forgot.
Yeah, animals and fungi are more genetically similar to each other than either are to other types of eukaryotes. They just have more genetic similarities. You could ask yourself the question of why is that? What does that actually mean? That's another question for another time. All right, so here's a question that will provide fuel for your thought between now and when we do our quiz on Friday. And I'm going to tell you right now, this question will be our short answer question on Friday's quiz. I'll just tell you this right now. Friday is not going to be 100% Quiz Friday, but this question is going to appear on Friday's quiz. And I'm going to expect that you know how to answer it. We're not going to do a lecture break and address this question, but I'm going to expect that you know how to deal with this. All right? Okay. These slides are on Canvas. Yeah, Emily.
Is it the what features do fungi share, or is it the how can we explain?
How can we explain? Yeah. All right. So here is just showing you the vegetative stage of a fungus. Here's the vegetative state existing as thread-like hyphae, and the hyphae come in two forms. They can be septate, where every cell is separated by a chitinous wall, or they can be what's called cenocytic, which means common, and they can all share a common cell. All right? So the hyphae can be structured one of two ways, but the thread-like hyphae is the norm for the vegetative state of fungi, as opposed to what stage? The reproductive stage. Good. Look at this. It's a Monday, and you're just rattling off, things that you've learned in the last few minutes. I love it. Happy Monday. All right, how can we organize fungi? This is the part that I know you want every single time we go through a chapter. This is a taxonomy. This is a taxonomy of fungi. So how can we organize fungi? What we do is we organize them based on reproductive strategy. So we don't necessarily organize them based on structure, although that plays a role in reproductive strategy. And we don't necessarily organize them according to genetic relationships, although that also plays a role in reproductive strategy. but we group them based primarily on how they reproduce. Okay? First group, Chytridiomycota. Chytridiomycota. And the answer to the question that nobody has yet to ask, yes, this will be on the exam and potentially on Friday's quiz. Okay? The ability to, I don't know, maybe match a phylum to a reproductive strategy. Just, putting that out there. So these can be single-celled or multicellular, and they have flagellated spores. At least some of them. Many of the species here have flagellated spores, both reproductive and asexual spores. And so if they have flagellated spores, where do you imagine you're going to find a lot of Chytridiomycotes? In water, right? Flagella work really nicely for moving through water, don't work so nicely for moving on land. Okay. All right, Chytridiomycota. So their reproductive strategy is they've got flagellated spores that aid dispersal. Zygomycota. So they tend to have sinusoidal hyphae, where all the cells are in some ways fused together. And another key word here is conjugated fungi. So what's common in zygomycotes is for two different fungal individuals to fuse together. Two fungal individuals to fuse together. And then when they do, these cells that have fused together form zygospores and sinusoidal or common cell hyphae. All right? We okay so far? So Chytridiomycotes, flagellated spores, that's unique to them. Zygomycotes, two fungal individuals fuse together and form zygospores at that point of fusion. Okay? You don't really find big, obvious reproductive structures in zygomycotes. The rest of our sponges, why am I calling these sponges? Is that the first time I've said it? Or have I said sponges several times? That's the first time, okay. In two of our remaining three groups, you have big, obvious reproductive structures. Ascomycota, ascomycotes. These are known as sac fungi. And that's because they produce these really large reproductive sacs that are called ascos, or ascus is singular, asci is plural. Okay, these large reproductive sacs that produce an enormous amount of sexual spores. ASCII is ASCI. And then I'll show you a picture and it'll have it written out. But these are fungi that produce a number of sexual spores, and then they tend to pop and release those spores all over the place. These are like, these types of fungi account for most of the fungal spores that you breathe in. You take a great big old deep breath, just know that there are several dozen fungal spores you just breathed in, and most of those are in this group, Ascomycota, and those are little baby fungi, ready to find a place to grow. It's wonderful. All right, so here we have a group of fungi known for their reproductive structure, these large asci or sexual structures with a number of sexual spores inside. Basidiomycota, known as the club fungi. These are mushrooms, okay? The mushrooms are a big structure associated with the reproductive stage of the fungus. So they produce a number of sexual spores as well, but rather than just filling them in this ascus, they fill them inside of this mushroom cap, okay, full of these reproductive spores. So these aren't going to shoot their spores out and fill the air with them. And so if you were going to disperse spores and you weren't going to disperse it by shooting them out, how would you do it? Think about mushrooms. What do we do with mushrooms? We eat them, right? And so you're starting to think of like angiosperms, right? Fill a fruit, entice animals. Animal eats fruit, spreads the seed somewhere else. That's this just strategy of a number of fungi in here. Basidiomycota. Okay? Attract animals to eat the mushroom, and as they do, they spread the spores out. Aid in dispersal, yeah.
So if they want the animals to eat them, why are some mushrooms poisonous?
That's a wonderful question. It asks you, are they poison? You have to ask yourself, are they poisonous to everything? And are there alternatives, right? Maybe it came from an ancestor that used animals to disperse it, but it disperses its spores just fine using wind, and now animals feeding of it is now disadvantageous. Yeah. So you have to ask yourself the question, how long have they been poisonous? How are they poisonous? And whom do they poison? Yeah. Just because they're poisonous to some animals doesn't mean they're poisonous to all. That's a good question. All right, so that's 4 of our 5. We only have 1 left. We're getting towards the end of your favorite part of every section. It's like when you get to the end of your bowl of ice cream and you're so sad to see it go, right? Here's that last bite. Glomeromycota. And these are just very, very unusual fungi when you compare them to the rest of fungi. They form these what you call arbuscular structures in plant roots. And what's really unique about them is they do not reproduce sexually. Sorry, get the rest of that screen on there. Okay, so of all of our 5 phyla, this is the only phylum that contains members that have not been found to reproduce sexually.
So it's asexual?
Yep, completely asexual reproducers.
Does that increase their diversity? Do I understand?
No.
No?
Yeah, no. Asexual reproduction leads to very little
asexually,
okay. Diversity.
So the 100 and 230 is actually a small number comparatively?
Yes. Mm-hmm. Yeah. I know it seems like a lot, but it's not all that much when you're talking about an entire phylum. For example, phylum Arthropoda in animals contains 1.4 million described species and probably another 9 million waiting to be named. Waiting for you, maybe, to go and name them. Not you? No. Okay, so here's an example of a chytridiomycote. And here you have little fungal individuals parasitizing. In this case, it's an arthropod, probably a copepod, and in here, parasitizing an algal species. Okay. That's kind of fun, right? Parasitic fungi. Why are they, if they're in chytridiomycota, what do you know about their sexual spores? They're flagellated. Where do you see these?
Water.
Parasitizing algae that live in water, parasitizing some kind of an arthropod that's in water. Look at that. It makes sense, right? Makes so much sense. Yeah, Cameron.
So all of these, they have the ability to asexually reproduce?
Yes.
That one of the colors
Does not reproduce sexually. Can it? I don't know. Does it? No. Yeah. Here's zygomycota, typical life cycle there. Here you get these individuals fusing. The fusion of these 2 fungal individuals and not producing a big reproductive structure. Remember, that was the characteristic of this phylum. No really large, distinct, and obvious reproductive structure. This is one thing we'll see in lab, bread molds. I've got, you know what I did is I had my kids put their nasty feet all over a piece of bread, stuck that thing in a Ziploc bag and put it in the bathroom, right? get a lot of moisture, allow the fungus from my kids' feet to just take root and grow all over that bread. You're gonna love it in lab this week.
Oh.
That's why you want to wear gloves, right? I just want you to know where the fungus came from when you are working through here. Here is Ascomycota. Here's this reproductive sac full of sexual spores. Here's the ascus, and then plural would be asci. Okay, now a large structure devoted to reproduction and to dispersal of spores. Here's it under a microscope. You can see an ascus, plural would be asci. Here's Basidiomycota, the mushrooms, right, club fungi, and then here's the reproductive life cycle or the reproductive cycle of a Basidiomycota. Here's your large reproductive structure. Yeah.
So you were mentioning the quiz and the test on Friday. We need to know what you showed us on the original slide, basically how they reproduce, not all these steps.
For which? The quiz or the exam?
Specified.
Well, so what I might do is I might, not for a quiz, but I might for an exam give you a reproductive cycle like this and block out like names like Basidium that would let you know, oh, that's Basidiomycota because it starts with the same, but just show you the typical stages and ask you to explain what phylum of fungus that is. That would be, I mean, there would be like a short answer question where you could leave it blank. You know, there'll tend to be 6 questions and you'll have to answer 4. So you just be like, not going to handle that, right? So sometimes, yeah. What I would more likely do is to be able to say, like, compare and contrast the reproductive strategies of Ascomycota with Basidiomycota, right? So then you can use whatever you know to do that, right? Yeah, one thing you have to learn is sometimes one additional point is not worth the time invested to get it, right? And I'm not saying don't do the absolute best that you can do. I'm saying you have to think big picture, right? And especially if we get to a time where you're doing a Gen Chem exam and an organismic biology exam on the same day, or you're doing an organismic biology exam and a OChem exam on the same day, right? For those of you that are in that, sometimes one additional point in one class is not worth, right? So you have to pick your battles, right? You have to pick your battles. That being said, the more you know from this, the more fuel you have to answer short answer questions. But as far as like a matching question goes, it'd be more like, you know, matching a phylum to like the typical, the typical reproductive strategy. Like for instance, you might expect to have like Ascomycota, Chytridiomycota, and Glomeromycota as 3 phyla, and then, you know, 3 different reproductive strategies, where one would be have a really large conspicuous reproductive structure. You're like, the only one on there that has that is Ascomycota. Basidiomycota also has it, but it's not one of the ones that I was asked to match. Right? Make sense? Okay, well, we did it. We made it through the first 3 framing questions. Nicely done, everybody. Give yourselves a round of applause. Very weak. The set. There we go.